So good morning and welcome to the North Central Region Water Network's Harmful Algal Bloom webinar series. My name is Eugene Brigg. I'm Program Director for Aquatic Ecosystems Extension at The Ohio State University, based at our School of Environment and Natural Resources, and I'm today's moderator. Welcome. This webinar is hosted by the Algal Bloom Action Team, of which Ohio State University, of course, is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the U.S. Team members include the National Network of Water Resources Research Institutes, say that 5 times fast, the North Central Region Water Network, and University Extension within each state in the North Central region. In addition to hosting our webinar series, our team is developing a website of resources, including fact sheets, frequently asked questions on harmful algal bloom topics, etc. I encourage each of you to visit our website, northcentralwater.org/HABs, which somebody will put in the chat presently. There you can explore some of the resources we have to date, including a What You Should Know fact sheet, a harmful algal bloom frequently asked questions database, and recordings from our previous webinars and our virtual harmful algal bloom research symposium, which is held each January. I should actually advance the slides while I'm speaking as well. Before we get started, just a few housekeeping items. We'll have a question and answer session after our speakers present today. Please type your questions and identify which speaker you are posing the question to in the Q&A panel. Put it in the Q&A panel. We will do our best to address all of them following the presentation. If there's a question already posted to the Q&A panel and you have the same or a very similar question, upvote the existing question, because we're going to prioritize those questions that have the most upvotes. Today's webinar is being recorded. It will be posted to the Algal Bloom Action Team website following today's session. If you have any technical issues or have questions about the Algal Bloom Action Team, please list those in the chat as well, and we will be happy to assist as best we can. So technical issues, questions about the general existence of the organization in chat, questions for our presenters in Q&A, please. Today's presentation is titled Lake Physics and Social Perspectives on Freshwater HABs. We will begin with a presentation by Dr. Adam Heathcote from the Science Museum of Minnesota. Dr. Heathcote is the director of the Science Museum of Minnesota's Department of Water and Climate Change and the St. Croix Watershed Research Station, where he leads a research team focused on the impacts of humans on our freshwater ecosystems. His research focuses on how human-induced global change can affect everything from prairie pothole lakes in the Midwestern USA to our most protected boreal and Arctic lakes. Dr. Heathcote received his PhD from Iowa State University, where he studied how harmful algal blooms alter food webs and biogeochemical cycling of nutrients, and was postdoctoral fellow at the University du Quebec in Montreal, where he studied the effect of atmospheric fertilization on primary production in boreal lakes. We will now begin. If you are ready, Dr. Heathcote.
Thank you, Eugene.
Certainly.
Just give me a moment to get my presentation up. Okay, so what I'm going to share with you today is the results from a study we did a couple years ago all across Minnesota, where I'm located. This was work that was funded both by the Minnesota Environmental Natural Resources Trust Fund as well as the Minnesota Pollution Control Agency. What we were really interested in looking at is a set of eutrophic lakes, which were known to have harmful algal blooms year after year, but then kind of drilling down into what might affect the interannual variability of those blooms, specifically looking at effects of internal loading and how physical measures of the lake, so things like thermal and oxygen structure, could be enhancing those blooms in certain years. So I always like to start these presentations with a very brief history of cyanobacteria. I think everyone here is pretty familiar with them, but I also like to say a couple nice things about cyanobacteria before I get into all the nasty things that we battle with them about. So as most of you are probably aware, they are among the oldest organisms on the planet, about 3.5 billion years old. They are responsible for the origination of oxygen in our atmosphere through the development of photosynthesis. And they also allowed plants to evolve by becoming the things that would then would become chloroplasts. Here in Minnesota, some of the oldest rocks that we have are actually fossils of cyanobacteria, things called stromatolites, which are fossilized cyanobacterial colonies. But there's more, of course, because of this long evolutionary history. They've developed all kinds of unique strategies that help them to be among the best competitors in the aquatic environments. So, for example, they are the only algae that can fix atmospheric nitrogen, and they do this through specialized cells. Not all cyanobacteria can do this, but a number of them can, called heterocytes. And these are small areas which have anaerobic environments that allow them to directly fix nitrogen from the atmosphere rather than relying on dissolved inorganic nitrogen in the form of nitrate or ammonia. They also can hibernate. So they're very resistant to poor conditions, and they have special strategies for surviving things like ice on in the winter. And they do this through resting stages called aconeites, which are actually specialized, very resistant cells that sit in the sediments or on the surface of the sediments, wait till conditions ripen in the next year, and are able to reform the colonies right away once conditions are better. They also take advantage of strength in numbers. So many cyanobacteria form very large colonies, colonies that are you're even able to see with the naked eye. And this allows them to do things like evade predation by zooplankton grazers. And of course, another reason we worry about them is they produce all kinds of toxins, what we call cyanotoxins. And these come in lots of different forms, and different cyanobacteria produce different types. So they're everything from neurotoxins, things that affect our nervous system, hepatotoxins that affect the liver and kidneys, and dermatoxins, which can cause skin irritations. And the reason they produce these toxins is actually still an area of scientific debate. Some of them may just be accidentally harmful to humans and wildlife, and others may be a strategy in terms of either evolving predation or actually battling against other algal groups. And of course, this is one of the areas that gets the most amount of press. So concerns for both human and wildlife health with these toxins. And something that'll be important for what I'm going to talk about today is they're also, unlike a lot of planktonic species, certain species of cyanobacteria are actually able to control their buoyancy. So that means they're able to migrate to different parts of a lake system. And so during the day when it's sunny, there's a lot of reason to be up near the surface so they can photosynthesize and take advantage of that light. But then at night, when that light is no longer available, there's no reason to be there. They can actually migrate into lower waters. And this is important because this is often where large pools of nutrients, especially phosphorus, hang out, and they're able to bring them back up with them within their colony when the lights turn on again. So those are all strategies of cyanobacteria, and what we're interested in is why they go bad, or why do they cause problems in terms of human and wildlife ecosystem health. And so I'm going to focus on two different questions as part of this. The first being, how do physical parameters, so things like the physical structure of a lake, the thermal structure of a lake, control both the timing and the management of internal loading of phosphorus in already eutrophied lakes. So internal loading means nutrients that are coming from within the system, so not nutrients that are coming from external sources. And do these internal sediment phosphorus sources fuel longer lasting and more toxic blooms? So is this an important thing to measure? So the potential for internal loading of phosphorus is highly dependent on redox conditions in lakes. This is data that was taken from core incubations, which were pulled from a lake and then brought back into the laboratory. And Bill James at the University of Wisconsin-Stout measured diffusive flux of phosphorus in both oxic and anoxic environments, and calculated that by just changing the redox conditions of the lake, you get like a 42-fold increase in diffusive flux. So this is pretty important when you think of large lake areas and over an entire summer season. So how might this affect harmful algal blooms? So in deep lakes, destratification as late season could be a possible secondary pulse of phosphorus. So you could get the spring flush of nutrients, but you could also get a secondary source when the lake mixes if there's a large amount of phosphorus building up in the Hypolineon throughout the entire open water season. And in shallow lakes where you may have continuous mixing, this may be a constant source of nutrients throughout the year. Now, importantly, the only way we can kind of look at these internal loading questions is through high frequency monitoring, because these things like polymictic mixing and these polymictic lakes may be on the order of just a few days. And so to be able to make sure we capture that, we need to have some way of measuring almost constantly. And we kind of have three different expectations for these different models. So if internal loading isn't important, we kind of see a basic seasonal bloom pattern where you get peak biomass and toxin production in the midsummer when nutrient concentrations are highest and temperatures is most optimal for cyanobacterial metabolism. However, if you have early mixing, so say if the lakes mix earlier in the late summer or early fall, you may see a double peak. So you can see that first seasonal peak and then a second one where those nutrients were all of a sudden made available that had been in the hypolineon before. And then in polymictic systems, you may actually see a step change where it just keeps increasing as more and more nutrients become available from internal sources. And so to do this, we looked at 5 lakes across southern Minnesota. So this is a primarily agricultural portion of Minnesota. It's mostly the tallgrass prairie biome. And we actually, as I mentioned, we put out buoys so that we could get these high frequency measures of temperature and oxygen. So these are measuring every 30 minutes throughout the entire open water season at approximately 1 meter intervals. These are the 5 lakes from the study. For this talk, for the purposes of time, I'm only going to talk about 3, but they are along a nice depth gradient. So we have a shallow lake, Pearl Lake, which has a max depth of just 5 meters, a kind of intermediate lake, Madison Lake, which has a depth of 15 meters, and then a deeper system, which has a max depth of 30 meters. We measured a whole suite of water quality parameters as part of this study. I'm not going to go into every single one of these, but the important ones I'm going to talk about today are chlorophyll A, so a proxy of algal biomass, 4 different cyanobacterial toxins. I'll focus just on anatoxin A and microcystin today, and then phytoplankton counts as well as all the major macronutrients. So our first example was that deepest lake, max depth of 30 meters, south center. So this is what we kind of think of as a typical dimictic lake. We'd only expect it to mix twice a year, once in the spring and once in the late fall. And in fact, that's what we see. Even by early May, it had already set up stratification. So just to orient you, these isopleph figures will all be the same. We have on the top the temperature profile. The horizontal axis spans an entire open water season from May through November. Horizontal lines on the top figure indicate stratification. So those are 1 degree Celsius isopleths. And then we have oxygen both measured at the top, so in the Hypolineon at 2 meters depth, and then in the bottom at 17 meters at this particular site. And you can see oxygen is rapidly drawn down to anoxic conditions, which are maintained all the way into November when the lake mixes over. And so how does this affect the nutrient concentrations? We see here both hypolimnetic and epilimnetic total phosphorus. Epilimnetic is in orange and hypolimnetic is in blue-green, and this will be the same for all the figures. So we see a moderate peak in total phosphorus that's seasonal in kind of the midsummer, huge amounts of phosphorus building up in the hypolimneon. So this is almost 350 parts per billion total phosphorus measured in the hypolimneon throughout the open water season. But because that mixing event happens, and I'll just say this dashed line here is when the lake turned over, so October 27th for this lake. And then we look at biomass, we see that algae kind of follow that typical seasonal pattern. Even though all that phosphorus that was built up in the hypolimneon did mix in late October, we don't see a response from the algae because the water was nice and cold. The photo period is quite short during this time of the year. So we don't see it as being a problem for this particular lake. So we see a large accumulation of total phosphorus. And I should mention that almost all that total phosphorus that we measured in the hypolimneon was actually SRP. So this is soluble reactive phosphorus. Peak algal mass followed, and toxins followed a kind of predictive seasonal patterns, and late fall mixing of the hypolimneon did not cause a secondary bloom. And you can see on the bottom panel here, I've added the 2 toxins I mentioned, microcystin and anatoxin. The red dashed line is just the drinking water standard for both those 2 toxins. And you can see that it kind of followed that typical seasonal pattern of the bloom in this system. So here's an early mixing example. So this is a lake of intermediate depth, max depth of just 15 meters. It's called Madison Lake, southwest Minnesota. And you can see it started off very similar to the previous example, already stratified when we deployed our buoys in mid-May. rapidly reached anoxic conditions. However, in this system, there was a huge storm that blew through in August with 60-mile-per-hour straight line winds, which prematurely mixed this lake. So instead of mixing in almost November, it mixed in August. And you can see what happens is once you mix a lake, even if it settles down again, you're never going to get that strong stratification because you've lost all that cold water for the rest of the season. So you can see it basically acted as a polymictic system for the rest of the season after that initial mixing event. And so how did this play out in terms of the nutrients and cyanobacteria? We saw a ramping up of total phosphorus following that mixing event. We lost almost all the phosphorus that was in the hypolimnion, so it was all transferred to the epilimnion after that mixing event. And we see this bimodal peak in algal biomass that accompanied it. So we see that first midsummer regular seasonal peak actually starting to decrease. And then that mixing event happened and we ramped up even further in terms of total algal biomass. And this is mimicked in all of the toxin concentrations as well. So these are the same two toxins, microcystin and antitoxin, showing that same pattern as algal biomass. So we see a secondary peak that we probably would not have observed in that year had that storm event not prematurely mixed this system. And finally, a shallow lake. So these are kind of one of the most interesting ones to me, because a lot of people, myself included, kind of assumed lakes, a lake with maxed out 5 meters is just pretty much mixing continuously throughout the open water season. There's no chance for anoxic hypolimnetic conditions because it's just so shallow. This lake is a huge bathtub, so very open. It's not something that would be sheltered that might set up stratification. But what we see is in the late summer, even just a 2-week or less calm period can actually lead to stratification because productivity is so high and biological oxygen demand is so high in this productive system. Even that small period of stratification actually leads to these anoxic conditions in this system. Again, this is a lake with max depth of less than 5 meters. And similarly, we see following the first stratification event, the largest increase in total phosphorus in the water column, as well as a response by the cyanobacteria. And this is also seen in the toxins. So just 1 week of anoxic conditions in the shallow lake was followed by peak bloom and toxin concentrations. So internally supplied phosphorus changes the timing and magnitude of these blooms. That was kind of the take-home message here. And so understanding how these internal loading dynamics work in some of these systems is going to be critical, especially as we think these things may change with climate patterns. So early destratification can lead to even bigger blooms that persist later in the year, and even shallow lakes can receive anaerobic diffusion of phosphorus under the right conditions. So weather is an important predictor of interannual variability in the size of blooms. Stronger summer storms are leading to earlier destratification, even in deep lakes, and prolonged calm conditions allow for short stratification periods where anoxic conditions can occur. And with that, I'll be happy to take questions at the end of the session.
Sincere thanks, Adam. I'm in this business because lakes are interesting to me, so I'm eager for the question and answer session to follow. So our second presentation will be by Dr. Katie Canfield from the U.S. Environmental Protection Agency. Dr. Canfield is a social scientist and the U.S. EPA's Office of Research and Development in the Atlantic Coastal Environmental Sciences Division in Rhode Island, USA. She's a qualitative social scientist by training, focused on applied research to improve stakeholder communication and engagement in water quality research. In the past, her work has focused on inclusive science communication and justice in tourism development. And hopefully we will begin the presentation. We had some technical issues with governmental machines earlier. Hopefully they are ironed. So welcome and thank you, Katie. I'm eager for your presentation.
Great. Yeah, we're gonna do our best to get the technology to behave here. And hopefully, please keep me posted if you can see what I'm seeing. We can see it. Yay. Awesome. Thank you, everyone. Sorry, I'm not used to presenting in Acrobat, so we'll do our best to please keep me posted, other panelists, if it's not working. But like Eugene said, I am Dr. Katie Canfield. I'm a social scientist based out of Narragansett, Rhode Island, and I'm going to be talking to you about the social perspectives side of this. What are the experiences of people who recreate at these sites that are prone to harmful algal blooms, as well as perspectives of people like yourselves, water professionals, in how we can improve awareness and risk communication around freshwater harmful algal blooms. So to situate how we do this kind of work as social scientists, our motivation is, while there is a lot of scientific research and biophysical understanding of HABs, there's limited research on how the users of these water bodies assess harmful algal blooms and the risk that they pose. So we know that environmental changes can impact people's perceptions, values, and uses of water bodies. So we wanted to know how about HABs specifically that might change their perspectives. And the goal here being that, or based also on the knowledge that managing and improving water quality can help requires environmental interventions and also public's buy-in to make those water quality improvements happen. So our goals with this research are to provide some signage recommendations to improve consistent language across sites, and to also hopefully better define what kind of research we need on the social side to understand HABs and human impacts, as well as shrinking that gap between what folks like yourselves know about HABs and what people who are recreating at HABs-prone water bodies know about the risks that these events can cause. Oh man. All right. So a little bit more about HABs and risk communication. So as you all may know, some of the constraints on monitoring and communication related to harmful algal blooms mean that for recreators, they often don't know about a bloom until after it's happened. So that can make it really difficult to assess the risk of going into a water body when there is an active bloom. So some of the challenges for risk communication here are all of the jargon involved in harmful algal blooms, and sometimes even like the terms of nutrients, nitrogen, all of those things can be difficult for just a random person trying to go for a swim to understand. The challenge of this both time and space-based disconnect between when we're polluting the system with nutrients and where and when the blooms may happen. So that can be difficult for our brains to understand how they're connected. And then also scientific uncertainty. We're very well aware that that's a big challenge to explain to people who aren't deep in HABs science every day, and how that uncertainty around the occurrence, identification, toxicity, and impacts of HABs, those all play into making risk communication really hard, which you again all may know. But there has been a lot of science communication research and environmental communication research that we think can help improve harmful algal bloom risk communication. And so that's where we started from in designing our research. So for this project, like I said, I'm based in New England, and so that's where most of our study sites were to begin with. Moving forward, that is going to expand across the country. Our team in this current research project, we're looking at a landscape scale, but we're focused on freshwater harmful algal blooms, and we're using a variety of methods, including content analysis of things like the previous presenter showed about dogs. So we'll do some analysis of what kind of language is used in those kinds of newspaper articles, things like that, and then also signage to understand what words are we using and how well are they resonating. We did key informant interviews with water professionals to understand their perspectives and methods of communicating with publics about harmful algal blooms. And then we did recreator intercept interviews where we basically get in between people who are trying to enjoy the water body and what they're trying to do. And we ask them a few questions about what they know about HABs and how that plays into their decision-making. And then we do qualitative coding to understand the major themes that come out of that. So since that may be less familiar to you, Oh, never mind. That's the next slide after this. So to put this into research questions, the 3 questions we were trying to answer or are trying to answer with this work are: what are key informants and recreators' perceptions of recreating near freshwater bodies prone to harmful algal blooms? How can we improve risk assessment and communication to recreators at HABs impacted freshwater bodies? And then also, how does changing water quality impact visitation and use of recreational sites with freshwater bodies that are prone to harmful algal blooms? So to lay the methods out graphically, we're doing 3 different forms of data collection for this project. So we did qualitative interviews last summer, and then a few more this summer. The ones this summer are occurring in Ohio, where we have a colleague working. We're doing use counts, so quantitatively counting how many people are coming to these different sites and how they're using the different sites. And then finally, that document review I mentioned, where we're looking at print, radio, social media, in situ posters about harmful algal blooms. And using all of those things, we'll be combining a variety of modes of analysis. So qualitative coding means looking for the themes based on what people are saying in those interviews. We'll be doing some basic statistics on those use counts, and then more qualitative and quantitative counting of what is said in the interviews. And then based on all of that, we're hoping to focus the analysis, and we focus some of the early analysis around what do we need research-wise, what communication is out there, what do people know about their exposure risks, what messaging works well and doesn't work well, what visuals should we use, and bringing all of that together to produce, our traditional reports and papers that scientists make, but also to provide some recommendations for water managers and professionals in how we can better communicate HABs risk. So for our project, for our recreator interviews, we focused on 6 sites. The top 3 were chosen in collaboration with biophysical researchers in our lab in Rhode Island, because they're doing monitoring work on these sites. So we were hoping to be able to combine the social and the biophysical data there. Those top 3 sites are all swimming sites, so that's a major draw. And they're all more rural sites, all in New England. And then the bottom 3, also in New England, are more urban sites. So we wanted to also see how use and perception differed across those different communities of people who live in more rural and more affluent communities compared to people who live in more city environments and might have more socioeconomic disparities and variety. So like I said, we did those 2 different sets of interviews. For the key informant interviews, we looked to people who would be potentially monitoring those water bodies where we did our recreator interviews, but also we have a colleague in Ohio, so she looked all over Ohio to see what those perspectives would be. So we have some interesting comparisons there that I'm happy to talk more about if you have specific questions. But looking across all of the interviews, what we found was some agreement across the board that monitoring has improved the documentation and general awareness of harmful algal blooms, but there wasn't a willingness to commit that these blooms have absolutely increased. So rather than maybe blooms increasing, it's better monitoring and documentation of the blooms that are happening. There was agreement that more money is needed, more funding is needed to sufficiently monitor all of the water bodies, and that there's a need to centralize all of this HABs research so that we can share our knowledge better, so that we can make better management recommendations and not recreate the wheel time after time. And another major theme that came up in the research needs conversation was better documentation and understanding of the human health impacts of harmful algal blooms. And I think that partially came up because we were so focused on recreator awareness. So that made us focus more on how is this impacting people as well. Oh, like I said, not the way I'm most familiar to present, so I'm sorry about that. So one of the questions that we asked in the interviews was, if you could communicate just one thing about HABs, what would it be? So this is people who lead lake associations, work for their state environmental management group, our researchers, what have you. And so if they had to just share one thing with people at these water bodies, and about managing them, they wanted to convey it's really uncertain. There is no one easy fix to get rid of harmful algal blooms. There are a lot of different drivers. There's nutrient loading, of course, and there's also other things that we are still trying to understand more of. They also wanted to convey that we don't always know exactly what the risks are for human health. So understanding the difference between the perceived risks that people may have compared to the proven risks, and we'll talk a little bit about those perceived risks in a minute. The other major thing that came up was the causes. So conveying that while we do have a lot of uncertainty around some of this, we also do know that nutrient loading is a major cause of the excess algal blooms, even if we can't always immediately tell if they're harmful or not. So communicating that the behavior change around nutrient loading would be one way we could all help with addressing HABs. Looking to what people who use these ponds say, we broke it down into a couple different sections. So these interviews ranged anywhere from 3 minutes to 30 minutes, depending on who the interviewer was and how chatty the interviewee was. But they covered basically why people came to these water bodies and what they knew about HABs and how increase in harmful algal blooms might change behavior. So looking at the terminology side of things, we interviewed approximately 150 people at these sites across last summer, and slightly over half of the interviewees had heard the term harmful algal bloom before. They were slightly familiar with this idea of toxic algae, but what that meant in practice and what that meant in observation and things like that, there wasn't a lot of confidence. There was limited familiarity with terms like harmful algal blooms, HABs, blue-green algae, and cyanobacteria. The ways that we heard it referenced across our interviews were very varied. So we heard people describe pond or lake water quality as being poor due to algae or green algae, toxic algae, or brain-eating bacteria. So there's definitely some communication gap there in what exactly a harmful algal bloom is. It's not something that is super well understood by people who are using these sites that are known to have HABs occur every single year. Looking at how their water use varied across these different sites. There was, of course, the scientifically proven and confirmed on the ground enjoyment and ease that comes with being in the outdoors and near water. For the urban sites, there was a sense of respite that the ponds and the parks provide for people, and the safety that it provides to be connected to nature. In terms of how changing water quality impacted use of water bodies, that really depended on how are they using these water bodies. For sites that are not for swimming, people didn't really care too much if the water quality was bad. They'd still go walk there if that was their go-to walking space that was not next to honking cars. But if they were swimming, they were potentially more concerned. We were able to document based on the recreators that they had observed, when there was algae, they did see that as a decline in the quality. So we happened to do a couple sets of our interviews when there was an active bloom. And so they were able to point out the swaths of neon green and say, well, that looks like bad water quality to me. There also are a lot of those aerators, those fountains in some of these water bodies, and the recreators were able to say, well, I think that that's helping to improve the water quality here. In how they assessed risk, it was primarily visual. So is the water clear? What's the water clarity like? How green is it? That's the main way that they were assessing their risk. And in terms of concern about the water, they were concerned about exposure basically by ingestion and immersion, not so much aerolized harmful algae. And then just general concern about risk of getting sick. They weren't super specific in terms of what they were concerned about, what that sickness might look like. And then again, the dog deaths. So the greatest familiarity we saw people have with the risk of HABs was related to dogs being exposed and then dying because of exposure. And on the communication realm, we found that there really was not at all consistent communication across our sites. As you can see, no two signs on this slide are the same. And these are from all of the water quality signs that we saw across the different sites. There was minimal awareness of the risk of harmful algae and the threat of it at the water bodies. Even when we were standing right in front of a warning sign, they would sometimes say, I've never seen a sign about that before. When we asked how people who did know about harmful algal blooms had learned about them, they said it was through newsletters from local pond associations or nonprofits, site-specific signs, and then radio stories were the main ways people had learned about harmful algal blooms. So those seem to be methods that work for certain populations. Again, I'm sorry for clicking somewhere I shouldn't. Something we were a little interested in was whether climate change played a role, and we did in perceptions of harmful algal bloom occurrence. We saw key informants said that they had observed a longer harmful algal bloom season with the warmer weather that's come in the recent years. Recreators had a kind of a local like folkloric awareness that later in the season we shouldn't swim in ponds. Warmer weather, warmer water means don't swim in the ponds, but not really a reason as to why that would be. And only one recreator explicitly said anything about climate change. In terms of looking a little bit at this dimension of environmental justice and the urban versus rural. In talking to the recreators, we unfortunately had a number of racist, primarily xenophobic comments to our questions about what don't you like about this site, what's changed over time at this site. And this was primarily a sense of ownership by some of the people that they were upset that there were people who didn't speak the same language as them, didn't look the same way as them, who came to use these sites in a way that they didn't approve of. So those weren't related to harmful algae, but did demonstrate division in the perceptions of the recreators and who's allowed. We saw that the urban ponds had significantly more use and more variety in the ways they were used. But there is, of course, a concern of subsistence fishing or recreational fishing in these ponds that have harmful algae blooms and what that can mean for human health. There were very limited translation of harmful algae bloom signage. One of our key informants told us they had to translate it themselves because it wasn't translated by the state agency. And then also something we were able to observe was that in the more affluent communities, there was more awareness of harmful algae blooms and the potential risks compared to in the more socioeconomically disadvantaged communities. So that emphasizes a need to target our communication towards the different communities in different ways. There aren't as many lake associations or significant communications in the more urban communities. And so targeting the communications towards the use and those language barriers would be very valuable there. The key informants focused on the importance of water quality being good for everyone. We didn't see any racism come up there, luckily, and recognize that importance of the refuge value of urban ponds for community members. Environmental justice did not come up explicitly, but we did talk to a couple water managers connected to an indigenous tribe, and they mentioned the importance of the cultural value of the water bodies and that being a major motivation for protection and use. So that being another piece of inclusive communication that we can tackle is considering the variety of meanings and importances that these water bodies can have to people of different identities. Based on this early work and early findings, some things that we will try to leverage into our signage recommendations looking forward, we're really at the write-up stage right now, is this concept of strategic ambiguity, something we all became a bit familiar with throughout the COVID pandemic, right? Science is always in progress. And so leveraging that in a way to say, this is the best that we know right now. And right now, the best choice is don't get in this water or something like that. And so leveraging that in our communication approaches, considering this idea of durability of message. So this comes also from communication research, knowing that just like we don't always notice like telephone lines in our neighborhoods, signs disappear to our vision as well. So changing our messages up every once in a while, leveraging different examples, nutrient loading, dogs, social norms, if we can all do it together to help make the message stick better. Adapting methods of inclusive science communication for effective risk communication. So that's knowing your audience, being very intentional, working with the community to understand why they care about the water body, and understanding their relationship. So you have effective communicators working with those different communities. When we talked to recreators, they pointed to the need for eye-catching visuals, and just another sign in a park isn't going to catch their eye if it's a bunch of words. So the need for creative visuals is important. And then context-specific communication feeds back into that idea of inclusive science communication, being aware of the different mental models and views of science that our different community members have, and considering their community concerns and uses of the water bodies. So like I said, this work, looking forward, we're looking to do more landscape scale analysis to focus on how HABs differently impact communities across the country, looking at if environmental justice communities are disproportionately exposed to HABs. Like I said, we did do use counts, so we're going to try to understand better how visitation varies throughout the season, and also combine that with data that we have from cell phone data on how many people are coming to these sites across time. And like I mentioned, also, we have a colleague in Ohio who is doing recreator interviews at Lake Harsha this summer. So we're learning a little bit about how the difference of a water body that's both drinking water and recreation, how that changes people's perceptions and knowledge and communication needs compared to our New England water bodies that are not for drinking water. So those are some more pieces that we're looking forward to, and that's everything for me. I look forward to your questions now, and there's my email if you have questions later.
Thanks for an excellent presentation, Katie. Now, I wouldn't be a good Ohioan if I didn't off the bat ask, who is your collaborator in Ohio?
So we have she's a contractor at EPA right now, but she's a PhD student. Sarah Goodrich is who we're working with. So
fantastic, thank you. Now, we have a lot of questions in the chat. Before we get to them, I'd like to thank everyone for participating in our Algal Bloom Action Team webinar today. Special thanks, of course, to our presenters who gave us 2 excellent presentations. I mean, really quite excellent today. Thank you. Also, thank you to Ann Nardi, of course, and Joe Bennell and their team at the North Central Region Water Network for hosting our webinar series and coordinating technical aspects. We couldn't do it without them. Following the Q&A session, please complete the survey at the link that I hope Ann will copy into the chat. We appreciate any additional comments or feedback you have for our series, and we will begin the question and answer session. Now, we have a lot of questions. I can't imagine that we'll get to them all before the top of the hour. Our presenters, would you mind typing your email addresses into our chat so that those who want to follow up that we don't get to can follow up directly? So without further ado, let me launch into our question and answers. So Katie, does your research also include homeowners who live near freshwater bodies that experience harmful algal blooms? I've heard that homeowners in these sites are against signage on HAB risks and may remove them in fear of decreased property values. How might this group be more effectively reached to communicate the risk of HABs?
Yeah, so we did ours based on active users of the ponds, which a lot of the time it came up in our conversation that they did live right around the sites that we were using, but we did not specifically seek out homeowners. For the people that we spoke to, a lot of them were aware of harmful algal bloom risks from lake associations. So I think that that is a really effective method. We also talked to leaders of lake associations about their involvement. And I think that that sense of community, like this is our pond, at least out here, that's a major way to motivate concern and desire to protect the water. So I think that leaning into that shared ownership and community protection is an angle. I understand that ugly signs aren't something that people want on their property, but I think that, again, that points to us needing to make targeted signs for different communities, but being consistent in our description of the harmful algal bloom, because that's where the confusion comes up. So I think, yeah, leaning into the lake association and that community ownership, and then also designing the signs with the community. So further leaning into that community ownership can help in making sure your message stays present for visitors.
Great, thanks, Katie. Adam, what can you tell us about the relationship between aquatic plants and algal blooms? If the aquatic plants are over harvested, will that contribute to algal blooms? Of course, that kind of ties into being a good limnologist, right? The work of Martin Scheffer is kind of classic in this field, but I'd like to hear your spin on this.
Sure, that's a great question. And actually, I didn't have time to talk about it, but one of the lakes in this study was a macrophyte dominated system. And we saw the complete opposite trend in terms of when algae bloomed, when nutrients were delivered to the system. So, you know, I always take it to heart to be aquatic plants' biggest cheerleader when I'm working on lakes and interacting with landowners and stakeholders, because I mean, I've seen with my own eyes the difference that you just described there, Eugene, between a plankton dominated and a aquatic plant dominated system. So this other lake in this study was called Lake Shakopee, and actually was one of the most impaired lakes in the state of Minnesota. But they did a whole bunch of work, dealt with some point source pollution, as well as a lot of non-point source mitigation in the watershed, and managed to flip it back to a aquatic plant dominated system during the study. And we saw no measurable planktonic phosphorus or chlorophyll throughout the entire open water season. And the entire lake was covered by mostly native, but some also invasive aquatic plants, which is a whole nother issue. And what was interesting was that in the fall, when turnover happened and all the aquatic plants died off, the phosphorus we measured in the lake was actually the highest among all the lakes in the study. So it wasn't that there wasn't phosphorus available. It was that it was all locked up in aquatic plant biomass. And so it was keeping it basically from the cyanobacteria.
Excellent. Thank you, Adam. Katie, are you aware of any interest in or work towards the creation of universal HAB symbol for signage, similar to a biohazard symbol? I guess a microcystin colony would, or a microcystin colony, right?
In my efforts to make fun slides to visualize this, I've had some challenge in coming up with one myself, but it absolutely is something that I think would be very helpful. I've talked with colleagues about it a little bit, and that would require a lot of different levels of approval, of course. But we do have a working group across the CDC and EPA and other government agencies that work on harmful algal blooms that work together. And that's definitely something that we've talked about, and I agree would be useful.
Fantastic, thank you. And Kristen has another question for Katie. What level and division of government would you recommend manage harmful algal bloom info and communication? In Minnesota, we have state, county, township, city, etc. At the state level, we have the Department of Natural Resources, Pollution Control Agency, Department of Health, Department of Agriculture, Conservation Districts, etc., all dealing with different aspects of water quality.
Yeah, absolutely. I agree it's challenging because there are so many different pieces of managing water quality. I think that having consistent signage at the state level is valuable because recreators will likely go to more than one site. But I think that having those different levels allows for how the communication about that sign to be, that allows for that to be more targeted. So when you do have that town level, you can, like we were talking about earlier with homeowners, be more aware of the specific uses and needs of that community, to share that knowledge that is consistent across the state, but share it in a way that's more effective for your specific towns. So I think that the different levels are all important. They just have different roles.
Great, thanks. Kristen again asks for Adam this time on Madison Lake. The microcystin concentration appears to have risen ahead of the chlorophyll A concentrations in the Hypolineon. Do you have any thoughts on this?
Sure. So that's something we've very commonly seen, and it's actually caused by the natural senescence, so the sinking of cyanobacterial cells into the Hypolineon. So actually, when we often see the most or the highest concentrations of dissolved toxins is after a bloom dies off. And that's because when a cell dies, it lyses and releases all the toxins all at once. And so we often see accumulation throughout the season of toxins in the Hypolineon, just as algae naturally sink and die.
Great, thanks. Now, Jenny asks, I assume for Adam, how would lake aeration impact algal blooms? And I'm thinking of a couple of different approaches to this, right? For deep water lakes, using aeration to destratify, of course, but there was a further question asked regarding aeration in shallow lakes. So could you kind of address that in a two-pronged approach to aeration and its usefulness against harmful algal bloom organisms?
So I didn't see the reference to the shallower lakes, but
that was in the chat. So I
okay.
glimpsed that from the chat.
So in general, oxic conditions will reduce the rate at which phosphorus is removed from sediments. So in that sense, it will reduce the potential internal loading of nutrients. But as I showed in the first example, if those lakes are stable and remain stable throughout the entire season, even the loading of phosphorus in the Hypolineon is not a large concern in terms of producing algal blooms, because when it is finally mixed, it's a time period, at least in the temperate zone where we are here, that isn't really a good time for algae to be able to respond to that input of nutrients. So yes, oxygenating the Hypolineon will reduce the amount of, or the potential for internal loading, but also it's a difficult thing to scale when you look at some of the sizes of some of these large lakes, and quite expensive as well.
Great, thank you. Adam asks, a different Adam, and again, I'm assuming of you, Adam, for these known bloomer lakes that were researched, are there lake management or watershed efforts to try to reduce the magnitude and frequency of these blooms? What practices or management approaches might be helpful to improve the performance of these systems?
Yep. So all of the lakes that I mentioned had some form of impairment on them, usually for phosphorus and chlorophyll. That's why we chose them, because we wanted to look at lakes that we knew would bloom, but wanted to look at interannual variance in blooms. And so a lot of the traditional types of mitigation or remediation efforts are in place. So things like trying to increase the amount of perennial vegetation on the landscape. Minnesota has recently, at least in the terms of my career, passed buffer laws that protect riparian areas. And so that work is always ongoing. But these are also located all within the most intensively agricultural portion of the state in southern Minnesota. And so, as many of you know, that's a difficult balancing act to maintain.
Great, thank you. I keep seeing questions appear in the chat as well. So a quick reminder, if you have a question for our presenters, type them in the Q&A panel or scan through that panel and upvote your question if it's already been posed, because we're going to prioritize and we certainly won't be able to get to all of these excellent questions for our excellent presentations. Lauren asks of Katie, did your research look at any differences in recreator perspectives among dog owners versus people without dogs?
Yeah, so we in analyzing the data, there definitely is more awareness among people who own dogs of what a harmful algae bloom is and the potential exposure risks for their pets. So that's, I guess, one difference. It was not something we were looking for. That's kind of one of those fun, surprising things that came out. Not fun, but consistent result. And then I saw there was a sub question about vets sharing that information about potential exposure to dogs. And they never shared explicitly. That was not something that was offered up, that they had learned about it from their vets, but that's absolutely something that some vets do share. I'm working on a paper about this project right now with someone who is a veterinarian, and that was one angle also of communication that we're going to recommend for dog owners specifically, is having that kind of information available in vet offices more visibly for dog owners to consider.
Great, thank you. Jeevan asks of Adam, are there any examples of early warning or prediction of harmful algal blooms? If so, how skillful are those methods? Also, would like to hear your insights in application of satellite products for harmful algal bloom tracking? And I can think of, obviously there are the predictive models that were developed for Western Lake Erie, which are pretty good based on nutrient loads. There are also, I know, some genetic indicators of the specific strands of DNA that are likely to produce toxins. I don't, any insight you have, I'm keen to hear.
Sure. So that was actually one of the things we're trying to figure out as part of this study, and it's still a work in progress. So as you mentioned, there are a lot of good predictive models for external inputs of nutrients and how that can be transferred into algal biomass. But one of the things we're working on, and it's still a work in progress, is how we can input some of these internal loading dynamics and systems where we think it's an important driver. So, there are a lot of physical lake models out there where this could be easily incorporated. What we need is more parameterization in terms of actual diffusive rates of phosphorus out of sediment. So how heterogeneous within a lake is that internal load of phosphorus, or the actual phosphorus in the sediments? How different is it lake to lake? And so that's something that we're looking at, collecting more data so that we can parameterize those existing models to be able to include this piece. In terms of satellite, the satellite products for observing and predicting blooms are great and wonderful, and I use them in quite a few of my studies. The only limitation, of course, is they only work on lakes of which are a large enough surface area to be able to be picked up. So that's something that will be improved as satellites continue to improve, but is currently a limitation, especially when you get to some of these smaller lake systems where an entire lake area might be included into a single pixel.
Great, thank you. Owen asks, did you observe any correlation or lack thereof between chlorophyll A and toxin production, or cyanobacterial cell counts and toxin production?
So we don't have cyanobacterial cell counts, but we do have both chlorophyll A and phycocyanin concentrations. And yes, both are very closely correlated. There's often a lag between chlorophyll A and cyanotoxin production. As I mentioned earlier, peak cyanotoxin production actually begins to increase as cyanobacteria biomass dies off. So we often do see this lag between the two, but they are correlated.
Great, thanks. I'm only going to get to probably one or two more questions. So once again, if you have questions that have gone unanswered, please write directly to our presenters. They're excellent and cordial. So Regan asks rather formally, Dr. Canfield, as a nonprofit who creates public-facing interactive web tools to help to map harmful algal blooms, do you have recommendations for funding or support avenues for improving on these tools, increasing visibility, and creating new tools?
Yeah, and combining this answer with the following question from Regan related to environmental justice. I think that, so the EPA has a ton of money going out right now related to environmental justice. And I think in the interest of creating more equitable awareness, that's one angle that I think would be valuable, and doing so in partnership between these wonderful scientific entities that know this work with community groups. I think that that's a big way to raise visibility, is to work with a trusted community partner that might not be as science heavy. And in terms of other funding angles, that's actually not something I've thought about a lot so far. But we also have like our EPA regional programs, and working in collaboration with the region and the science sides of things can be a great opportunity. So I'm happy to email back and forth more about that, though. A good question.
Great, thank you. And I'm probably going to take just one more question unless you advise me to do differently. Feel free to interject in the chat or verbally, but I really appreciate these excellent presentations and your excellent address of the questions to follow as well. So John asks, can you please put up a link to the 5-lake study for us to download and read, which is kind of a quick one-off, so I'll let you paste that into the chat as you're available, and then I will go on to Mark's question. Is your shallow lake actually a flooded marsh? We have many of these in Wisconsin, with vast portions of the lakes actually would have been rich fertile meadows and riparian wetlands pre-settlement before dams were installed to raise water levels.
Yeah, so this is actually, I think you're referring to Pearl Lake, which was the shallowest one. And this was a prairie pothole lake. So it's not necessarily a flooded marsh. It's a kind of really typical round bathtub lake where a block of ice sat for a couple thousand years and formed it.
Great. Thank you. Well, we're a bit more than an hour in. So Adam, I don't know if you have a link to the study that was requested, but if so, if you could post it into the chat quickly. If you don't have the opportunity to, John, please write to Adam directly. Thank you so much for your time. Thank you for all who participated today. It was an excellent session. Thank you.